Okay, so another essay plan that we're going to look at for biopsychology. Same caveats apply. Don't just memorise this off by heart. Make sure that you understand it so you can apply it if there's a particular scenario that's provided, or if the wording of the question means that you have to tailor it into a very specific sort of way. So if you think back to the mock you did where it asked to discuss Pavlov and Skinner's contributions to psychology and understanding of human behaviour, if all you spoke about was Pavlov's dogs and Skinner's rats, then you were limited to the first two kind of boundaries on the essay. So do read every question carefully and really think about what it's asking you. Okay. So hemispheric lateralisation, first key thing you want to do is not confuse this with localisation. You want to provide a definition to demonstrate to the examiner that you're really clear on that. And it's the idea that the two hemispheres have specialised functions and are not exactly alike. So they do have independent roles, they work separately from each other. So in this one of the first things you could talk about again is the idea of Baroka, who found exactly the same damage on the right side of the brain had no impact on learning ability, whereas on the left side in this particular area, which then became Baroka's area, there was a huge impact on language. So for example, Tannen experiencing expressive aphasia. And this led Broca to start thinking not only about localisation but also lateralisation as well. So the idea that the left hemisphere specialises in language production and language processing, as we find out later on, and supports the idea of lateralisation, that each hemisphere is specialised in a particular role. Okay. The two hemispheres are connected by the corpus callosum, which is mentioned on the PowerPoint slide, and it allows the two hemispheres to communicate between the two. And then you could go on to talk about split brain patients. So of course split brain patients are patients who receive neurosurgery to reduce epileptic seizures, which can be life-threatening. So although split brain surgery can also have significant impact on the way that people can function, that's not as significant as the other option, which potentially for some people with very severe epileptic seizures could be death. Okay, it's also a side effect of this, as well as reducing epilepsy in certain people. It allows psychologists to observe the role of each hemisphere and how they work independently of each other. So they are very useful from an experimental point of view. Although, of course, ethically we wouldn't be allowed to create split brain patients just for the benefit of studying them. And then you would go on to talk about Sperry's research as well, which I will talk about in this slide, but you do also need to know enough about this that potentially it could be a separate question. It could be describe and evaluate Sperry's research into hemispheric lateralisation that you would need to talk about. If that was the case, think very, very carefully about that one, okay, because it's very unlikely that at A2 all you would have is a very simple describe and evaluate research conducted. So we know, as you know, you had a control group, an experimental group, and a control group of 11 split brain patients who had been given this procedure because they were experiencing epilepsy, and this was supposed to reduce that epilepsy. So if we have, the procedure was to place participants on one side of a table, and then the way they were able to observe items is they were only you're only able to perceive them in a certain field of your vision. So they were asked, split brain patients and the control group were both asked to focus on a dot that was in the middle of their sight. They had to look at that and they weren't allowed to look away from it. However, things were flashed into the left and the right visual field to allow them to explore the connection between visual processing, verbalizing, so language production as well, as well as many, many other things, so everything to do with hemispheric lateralisation. So here you have an example of a non-split brain patient. So this is what would happen to someone with a non-split who hadn't been through the procedure. If they were part of Sperry's research, they'd be asked to focus on a dot right in front of them, so they shouldn't look left or right. So the word key would be flashed up in the left visual field. Because it's in the left visual field, it'd be processed by the right hemisphere. A psychologist would then say, can you read the word that was flashed into the left visual hemisphere, please? into the left visual field. And in a non-split brain patient, they would the information would cross over the corpus callosum. They'd be able to provide the visual information to the language centres on the left side of the brain, so it would cross over, and they'd be able to say the word key. Split brain patient, who was shown that the word key was flashed up in the left visual field, would the information, exactly like the non-split brain patient, would be processed in the right hemisphere. The psychologist would ask them, can you tell me the word that was flashed up in that you saw that was flashed up in your left visual field? And because they weren't able to communicate, their two hemispheres weren't able to communicate, then they weren't able to answer that, they weren't able to verbalise it. However, a key in the right visual field would be processed in the left hemisphere visually, and then of course the language centres are also in the left hemisphere, so the person would be able to say key when they saw that. Same thing happened, they were shown an image of a key, and then they were asked with their left hand to try and find that key. So if the key was flashed in the left visual field as a non-split brain patient, they would process it. But because they were using their left hand is controlled by the right hemisphere, just like which is the same hemisphere that is processing information from the left visual field. So the image is flashed from the left visual field, processed in the right hemisphere, and then doesn't even need communication, but then the right hemisphere tells the left hand to search out, so they couldn't see the objects, they had to search out and find the one which felt most familiar to their object that they'd been shown in the left hemisphere, and they were able to, if I can get my computer to work, find the key. So they were able to do that in a non-split brain, just as anybody who hadn't had the surgery had been able to. Then we had the key in a split brain patient. The key was flashed into the left visual hemisphere, processed by the right hemisphere, flashed in the left visual field. Processed in the left, seen in the left visual field, processed in the right hemisphere, and then because it was the left hand, then the right hemisphere was still controlling that, so it was able to find the key which matched most closely. So that was all fine. The problems came when the key was shown in the right visual field, which was processed by the left hemisphere, but then the person was asked to find the key with the left with their left hand. In a non-split brain patient, the corpus callosum would transfer that information across into the right hemisphere, but that's not possible with a split brain patient, so they didn't do that. So they weren't able to, they just couldn't identify what the key was. They could see the key and they could process the information in the left hemisphere, but because they needed it to cross over the corpus callosum so that the right hemisphere could tell the left hand to find it, they weren't able to do that. Okay, and then we have Sperry here, who conducted research, so showed them the word key on the left, which is processed in the right hemisphere, and then they were asked to find a key with their left hand, which they were able to do, or they were shown the word ring in the right hemisphere, and they were able to say the word ring. But of course, if those two tasks were swapped over in a split brain patient, they wouldn't be able to do them. Okay. So these are the evaluation points that we will have a quick look at for hemispheric lateralisation. They're much broader. There's a variety of different things we could talk about, and then we'll also go through the evaluation points for Sperry and have a quick look at how you'd have a much shorter essay response to Sperry's research, but it would still allow you to access the higher marks. It's very unlikely, unfortunately, that there would be a 16-marker describe and evaluate Sperry's research, which is a bit of a shame, but there could definitely be a smaller mark question on it, so it's very useful. And of course, you could use this to talk about your AO1 in Sperry's research for hemispheric lateralisation. In AO1, discuss Sperry's procedure and his findings. Okay. And then in AO3 you can go into explaining why they support the idea of hemispheric lateralisation. So be very careful to, in Sperry's research, keep the clear defined line between AO1 and AO3. But you do need Sperry's research, the procedure in AO1, so that you can talk about how it supports, his findings support the hemispheric lateralisation in AO3. But then you can go on to talk about methodological issues. If you don't talk about Sperry's methodology in AO1, then you can't do it. You can't evaluate it in AO3. Okay. So, one of the first strengths of hemispheric lateralisation is it's been found to increase neural processing capacity. So the example I've got in my notes is that if one hemisphere is visually processing a face, thinking I recognise that, where does it come from, accessing your memory banks, the other one's available to process the verbal response that you want to make. So you see somebody that you recognise, one part of your hemisphere is trying to remember who they are as kind of going through lots of different faces trying to match them up, while the other hemisphere is making polite conversation as well. So you can do more than one task. You can multitask within your brain with the two hemispheres. And Rogers, a lovely piece of research, found that lateralisation gave the domestic chicken the ability to both search for food and remain vigilant to predators. So one hemisphere was searching for food, thinking about, oh, where's the best place to look? Is it under this bush? Is it in these trees? Whatever it might be. Whatever chickens think about food. And then remain vigilant for predators. So the whole time the other hemisphere would be able to be kind of thinking about, now is that a noise? Is that a predator? Is that something? Do we need to run yet? So whereas if you remained vigilant for predators the whole time, the other hemisphere, and that was across the whole of your brain, and your other hemisphere couldn't look for food, you'd die because you wouldn't be looking for food. You'd constantly be worrying about predators, or vice versa. If you were always looking for food, you'd be a very, very fat chicken, but you wouldn't last for very long because predators would eat you because you hadn't been looking out for them. Which suggests that lateralisation is beneficial for our survival. So therefore, a strength of lateralisation is it can be supported by the evolutionary approach. So you could talk about the idea that maybe it has some sort of concurrent validity or some validity of sorts because it is supported by other theories which add credibility to it. However, a problem with that approach is that it has been found that hemispheric lateralisation has some physical disadvantages. So the fact that our brains become lateralised, i.e. Each hemisphere kind of controls various different things, causes us problems later on. And you've got Tonston, who found a small but significant relationship between handedness and immune disorders. So if you were either left or right-handed, and I think unfortunately might be wrong, but I think it's left-handed, then you are more prone to immune disorders, or I suppose various bonds possibly. And the idea of how this is linked is handedness is influenced by which hemisphere is dominant. So of course if you're you'd be your right hemisphere, and if your right hemisphere was dominant, you'd be left-handed. If your left hemisphere was dominant, you would be right-handed as well. So this suggests that the same genetic process that led to lateralisation may lead to health conditions. So the same process of evolutionary process almost, or the same process that led us to develop lateralisation has also led to health conditions, and it's a weakness because then it goes against the evolutionary approach, suggesting that lateralisation isn't beneficial, it's not advantageous, so it shouldn't have been something that was developed, or if it was, it's not supported by the evolutionary approach, so removes that concurrent validity which we provide. Okay. Supporting research, so you could then discuss what Sperry found, so the idea that if Key was shown in a certain visual hemisphere, then at certain times visual split brain patients couldn't identify it. So if the word key was flashed up in the left visual field, it was processed in the right hemisphere, therefore because they couldn't send the information across the corpus callosum to the left hemisphere to verbalise what they were seeing, they weren't able to provide that answer. That of course all supports lateralisation because it supports the idea that the two hemispheres work independently and communicate via the corpus callosum. So it indicates that language isn't on both sides of the brain. So the hemispheres do have specialised roles. However, there are some problems with Sperry's research. So the sample was made up of 11 patients who had previously suffered epilepsy. So that causes two problems. First of all, they're very specialised. They're a very small specialised sample, so small 11. So you've got to question the population validity there. And secondly, they're very unique. The fact they'd had to go through all these processes, but most importantly, epilepsy could be viewed as a confounding variable. So the idea is that confounding variable is something that systematically varies with the IV. So if it's present in one condition and not in the other, and it isn't the IV, then it's confounding variable rather than extraneous variables, which are more at random. So some people in both conditions might be different ages and things like that, whereas this a confounding variable varies systematically. And the fact that some people, not only were they split brain patients, which should have been the only difference between the two, but some of the participants in split brain, all of the participants in split brain had suffered with epilepsy, and none of them in the control group had. So that was another participant variable that could act as a confounding variable, because of course epilepsy could, just by having epilepsy, being born with it, or sort of however it develops in itself, there's an element of brain. There must be an element of faulty brain processing there, or the brain works differently to the people in the control group. And secondly, seizures can cause brain damage as well. So that would be something else that wasn't controlled for, that could be a confounding variable. And then another issue, an extraneous variable, would be the fact that all of the patients had different varieties of to how much their corpus callosum had been lesioned. So the more lesioned it was, the more significant the difference might be. So they weren't all comparable either, and that wasn't controlled for. So all of that makes it difficult to generalise to the wider population, and also suggests that the research lacks clear cause and effect and reduces the internal validity, because there's always confounding and extraneous variables reduce the validity. Another weakness or another problem is that the original findings haven't since been supported, and you've got a case study of JW who found that he developed the capacity to produce spoken language using the right side of his brain. And then Kim Peek, who's a really, really interesting example. He's the person who inspired the character of Rain Man. Was born without a corpus callosum and developed the capacity for language processing and production in both hemispheres, which allowed his eyes to process written information independently of each other. So he developed, essentially, not quite like this, but he developed the Broca's area and Wernicke's area in both sides of his brain, so his brain compensated and allowed him to process that information. So because they worked independently, he could read 2 pages of books independently and process the information. So the book from the left-hand page of the book would be seen visually in the right hemisphere and processed and understood in the right hemisphere, and the information from the right page would be processed visually and then understood, the language processed and understood in the right hemisphere. So he could read 2 pages simultaneously and understand both of those pages. And of course that's a weakness, because it appears that replication of Sperry's research was unsuccessful, and therefore calls into question the reliability of Sperry's research and the conclusions drawn. Could be how you'd summarise that.